Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as video is getting uploaded. Okay guys, don't forget to subscribe. Okay, we shall start today's session. In today's session we are going to discuss the last topic of reproductive health, that is infertility. Okay, about infertility and assisted reproductive technologies, we are discussing about that one in this session. Okay, we shall start with this infertility. With this infertility we'll get on page number 63, which is something like this in NCERT textbook. Oh, fine. Now we shall start with this infertility. First we shall see what is infertility. Okay, infertility, it is inability to conceive or produce children even after 2 years of unprotected sexual cohabitation. Okay, it is inability, unable to conceive or unable to produce children even after 2 years. Even after 2 years of unprotected sexual cohabitation. Unprotected sexual cohabitation means without using contraceptives. Any kind of contraceptives, might be condoms, might be barriers, might be IUDs, natural barriers, natural method, or any of those. Without using any of these contraceptives, then also if a couple is unable to produce children, then we call it as that individual is infertile or it is leading to infertility. This will be a definition for infertility. What are the reasons for infertility? It is physical. Physical means that physical appearance, that is body of if it is not properly developed. Congenital, that is chromosomal. Diseases, any diseases it might be. Some of the diseases are going to affect the production of gametes also. Drugs here not only represents that cocaine, heroin and all. Obviously these are there. With that, even tobacco, which is used in the form of snuff or which is smoked, that also is going to affect the production of gamete. Immunological, that is their immune system, if it is weak. Even psychological and even mentally, if they are not fit, then their production of gametes will be reduced. If gametes are not produced, then they are unable to reproduce. Okay, so what is infertility? Mention the reasons for infertility. We have got this question for 2 marks, so you must know this definition and the reasons for it. Now, as you can find in the society or in your surroundings, many of the couples are not having children. Now, in that couple, female will be blamed. Hope you might have heard about this or you might have seen. Okay, more often females are blamed, but the problem lies in the male partner also, not only in female, even in male. Okay, for this special healthcare units are available. Infertility clinics are available, which can help you to diagnose correct treatment of some of these disorders and enable this to have these couples to have children. Okay, so for that we have assisted reproductive technologies. The couple who does not have who are unable to have children, who are suffering with the infertility, they can go through this assisted reproductive technologies which help them to have children. Okay, how is it possible? We shall see. The first technique here which has been mentioned, that is IVFET. What's its full form? In vitro fertilization embryo transfer. This in vitro fertilization embryo transfer, it in that most important is test tube baby program. It is commonly called as test tube baby program also. Now this test tube baby program, by the name, don't imagine directly that there is a test tube in that a small baby, finger size baby is present. No. Why we call it as test tube baby program? Because it has been developed, that baby has been formed in the laboratory. For that reason we call it as test tube baby program. Okay, we are using intrauterine transfer, in vivo fertilization. These terms we are using in IVFET. Next, second one we have ZIFT, Zygote Intra-Philoppine Transfer. Third one, Gamete Intra-Philoppine Transfer. The next, fourth one, Intracytoplasmic Sperm Injection. Then we have AI, that is artificial insemination. Okay, in this we are using one of the term called as IUI, Intrauterine Insemination. Now, in these are the 5 assisted reproductive technologies. All 5 are very important. 5-mark question it can be asked, and it has been asked. So listen carefully to that. So I will explain that one here. First, starting with the IVFET. Okay. Now first one we are saying it as IVFET. What we said for IVFET, that is in vitro fertilization embryo transfer, which means in vitro. First we are using a term called as in vitro. What do you mean by in vitro? In vitro means something which takes place in the laboratory or in an artificial conditions. For that we call it as in vitro. Okay, if we are using in vivo means it is in natural conditions, right? In vivo means which takes place in a natural conditions. In vitro which takes place in the laboratory or in an artificial conditions. In vivo, natural conditions. Now in vitro fertilization. Now fertilization is taking place in the laboratory. Now as you know, fertilization is what? Fusion of male and female gametes. Where does this fertilization takes place in human? That is in the female reproductive system. Where in the female reproductive system? It takes place in the fallopian tube, right? But here that fertilization takes place in the laboratory. What we are doing is in vivo fertilization. Here the ova, that is ova or egg, it has been collected from a wife or donor. Okay, ova, it is collected from a wife or a donor, from a female, and sperm, it is collected from husband or donor. Okay, if husband is able to produce sperm, then we can collect from that. If he is unable, then we can go to the donor. Many of the donors are available, will donate their sperm. And if wife is able to produce a gamete called as egg, then we can collect from her. If not, we can get from donor who is ready to donate their egg. Okay, now this we have in our laboratory in two different test tubes. You can imagine like this. Now we are taking these two, ova and a sperm, and making it to get fertilized to form zygote. Okay, now this sperm and ova is getting fertilized to form a zygote. Now we are forcing that one stimulated in the laboratory, providing natural conditions as it will be provided in the mother's body, that is in the female reproductive system. What conditions will be present in the fallopian tube, that conditions will be provided here in the laboratory, so that easily fertilization can take place and it leads to the formation of zygote. Okay, now this zygote which has been formed, that can be this zygote, where it will be formed in a female reproductive system. Actually in female reproductive system, fertilization takes place in a fallopian tube and zygote is formed in a fallopian tube, right? This is actual location. But when we are doing this one in the laboratory, it takes place in the laboratory in a petri plate, you can imagine. Now when zygote has been formed, this zygote you can take and you can introduce in the female body. Okay, any of the female, that she might be a wife or a surrogate. Okay, she might be a wife. A wife means whose egg it was, she, or it might be a surrogate. Now this zygote is taken. Now female reproductive system, it was something like this, right? So here zygote will be formed. Now through the vagina, this zygote will be introduced into the fallopian tube. Now if zygote is introduced into the fallopian tube of a female, then we call it as ZIFT. This process we call it as ZIFT. What is that ZIFT? Zygote intra-Fallopian transfer. Zygote is transferred inside the fallopian tube. Okay, zygote is transferred inside the fallopian tube. Now not only zygote, even when it has started its cleavage. As we know, zygote will start its cleavage forming 2 blastomeres, 4 blastomeres, 8 blastomeres. Less than 8 blastomeres, either zygote or less than 8 blastomeres. Okay, either zygote or less than 8 blastomeres embryo can be transferred inside the fallopian tube of the female who is willing to have that. Okay, so now this one we call it as zygote intra-Fallopian transfer. Either zygote or less than 8 blastomere, if it is transferred, we call it as ZIFT. Okay, but we were discussing about IVFET as per you remember. Still we have not completed IVFET. Now in some cases zygote is transferred. In some of the cases zygote is present in the lab only, that is in the petri plate, and this zygote will continue its further division. What division? That is this cleavage, forming more than 8 blastomeres to form an embryo. Okay, now this embryo will be having more than 8 blastomeres. If a embryo of more than 8 blastomeres is getting transferred into the female reproductive system, then it will be transferred into the uterus. This is the uterus, right? So this one will be transferred into the uterus. Okay, if embryo is transferred, then that embryo will be transferred in the uterus. If zygote or less than 8 blastomere embryo is transferred, that will be transferred in the fallopian tube. Now when embryo is transferred, it is in vitro. In vitro fertilization embryo transfer. Fertilization took place outside the body and this embryo has been transferred into the female reproductive system, that is IVFET. Okay, now when it has been transferred in IVFET, here we can use a term called as IUT, that is intrauterine transfer. Intrauterine transfer. Now this intrauterine transfer means what? Intra means inside. Inside the uterus, it has been transferred. What has been transferred in the uterus? Either zygote or embryo. Embryo has been transferred. For this a transfer we call it as IUT, intrauterine transfer. Hope you understood this. Okay, so this one, the one who is unable to produce a gamete, or one who wanted to go with the natural process, they can proceed with this. That is IVFET. Ova is collected a wife, sperm is collected from the husband, zygote is formed in the laboratory. Zygote can be introduced in the fallopian tube, or less than 8 blastomeres embryo can be introduced in the fallopian tube, and it will continue its further development in the female reproductive system as normal it does in the natural fertilization. Or we can go with the next step. Zygote can be allowed to undergo further step in the laboratory only, and that zygote will convert into an embryo of more than 8 blastomeres. If embryo of more than 8 blastomere is getting transferred in the female reproductive system, then it should be transferred into the uterus, not in the fallopian tube, because in natural conditions that zygote is formed in fallopian tube, whereas embryo will be formed in the uterus. So taking a consideration of a natural ecosystem, natural development, that we are using it here in the artificial medium also. Okay, so this is about IVFET. Now in vitro fertilization we said in laboratory, in vivo we said in natural conditions. Next, here we have covered two. One is IVFET, next one is ZIFT. Both have been covered here. Next I shall proceed with the GIFT, third one. This was second one, third one. GIFT. What is GIFT? Gamete intra-Fallopian transfer. Now with this only you will come to know that what we are going to discuss. Gamete intra-Fallopian transfer means gamete is transferred into the fallopian tube. Okay, transfer of an collected ovum from a donor into the fallopian tube of another female who cannot produce one. Okay, but providing suitable conditions. Now here we can find there is a female. Okay, female donor. Female donor, what she is donating? She is donating egg. Now this egg is collected from a female donor and it is transferred into the fallopian tube. Into the fallopian tube. Okay, into another female who could not produce this egg. Okay, could not produce egg. Okay, female who can produce egg, she is donating her egg. We can collect that egg, take a egg and transfer it in the fallopian tube of another female who could not produce egg, but she can provide suitable environment for fertilization and further development. Okay, environment she can provide. What environment? Uterus. Okay, a fallopian tube, it is normal, but one defect she has, that she is unable to produce gamete, but further development it can happen in her body. So this case can take place, and for this we call it as gamete intra-Fallopian transfer, because gamete is getting transferred into the fallopian tube. Okay, next, fourth one, ICSI, intracytoplasmic sperm injection. Okay, intracytoplasmic sperm injection is nothing but it is a specialized procedure where sperm, okay, we are taking sperm, it is directly injected into the ova. In ova, we have said it consists of ooplasm, or it is also called as cytoplasm. Hope you remember this, right? Sperm is directly injected into the ooplasm, which means that fertilization is taking place in the laboratory. Okay, if a male is unable to produce large number of sperms, large number means 350 million number of sperms. Okay, 350 millions of sperm should be produced in per ejaculation. If less than that, if he is able to produce, then we can, because if it is less than that, then that individual is infertile. For that reason, what we can do, we can collect this sperm or from the husband or a donor, and we are collecting the ovum from the wife or a donor, and we are taking that sperm and directly we are injecting that one in the cytoplasm of the egg. Okay, cytoplasm of the egg, and fertilization takes place for the development. Either you can transfer this zygote into the fallopian tube, or either you can transfer embryo into the fallopian tube, as we have discussed earlier. For this, we call it as ICSI, intracytoplasmic sperm injection. Okay, next, fifth one, artificial insemination, AI. Artificial insemination, the name itself shows that is insemination could not take place if an infertility cases, either due to inability of the male partner to inseminate or female, or due to if there is a very low sperm count in a male. Okay, very low sperm count in the male, then what we can do, we can collect the semen from that male. We are collecting semen here, and that semen is ejaculated artificially in the vagina of the female. Okay, in the vagina or uterus of female. Now this semen can be collected from the husband or a donor, as I said earlier, because he is unable to ejaculate or unable to inseminate. So because of that reason, we are collecting semen artificially. We are introducing into the vagina or uterus of the female, so that, as we can find, it is something like this, right? Now even ejaculation takes place, insemination takes place, that will take place in the vagina. Now here either we can introduce in the vagina or into the uterus. Now these sperms, when it has reached to the vagina or uterus, what they'll do, they'll swim to the ova where it is present, and it can get fertilized naturally. Okay, that is artificial insemination, right? When sperm count is low, in that case we are undergoing, we can choose artificial insemination. In this, we are using a term called as IUI, intrauterine insemination. Intrauterine, intra means inside, uterus, insemination, we are inseminating sperms or semen in the uterus, so it can be called as intrauterine insemination. Okay, so these are the few techniques which can be used for couples who is unable to have children. Okay, so these are the first one is IVFET, second one is ZIFT, third one GIFT, ICSI, and AI. After this, here these methods, these techniques are there, but you find in your surroundings many of the couples are struggling to have children. They are having lot of problems related to having children. So how is that possible? Why they are not using all these techniques, ART techniques, right? So the question is that these ART techniques are available, but it is benefited only for few. What are the reasons for this? First reason, you can say these techniques require extreme high precision handling by specialized professionals and expert instrumentations. Okay, techniques are there. It requires high precision handling by specialized professionals, and it can be handled carefully. This is the first one. Second one is that these facilities are presently available in very few centers in the country. Okay, these facilities which you find, these are not present everywhere. As you find everywhere hospitals are there, then every hospital is able to treat this kind of disorders. There are special centers, special clinics are there, infertility clinics are available, which is not there in every part of this country. But nowadays, as per we have seen that infertility clinics are there, at least per district one we can find that. So that is available. Next one is emotional, religious, and other factors are also deterrents in the adoption of these methods. There is an emotional, religious, and other factors are also responsible, so that they are not willing to adopt this method. Okay, they are not willing by themselves. Second one is they don't want it to, it is not present everywhere in the country. Third one is that it requires high precision, which means it is highly costly. So when it is costly, then many of the people could not afford it. If they could not afford it, then they won't go for this method, right? So for that reason, these are the three reasons why every infertile couple could not have children. Okay, and there is a unawareness also sometimes. Okay, these are the three reasons why it is benefited for few, not for all. Now, with this, what we have studied, its main aim is what? That is to have children. But according to my suggestion, there are many of the orphanage, there are many of the destitute children available. We can go for that and have those children, because their growth, unless they are taken care of till maturity, their survival rate has been reduced. So when we have an option to have a children, rather than spending so much of money with going with a lot of lengthy procedure, so that money you can spend it on the other children and bring it home and grow it like your child, and it can be helpful for others also and even for you. Okay, fine. This was some information related to infertility, what we have discussed, what I have mentioned here about assisted reproductive technologies. These are its full form. If you have not copied, please pause the video and copy all that information. Now you might be having a question that why, from where you are going to get the notes. The notes you will find on page number 64, okay, of your textbook. Completely in detail they have given about assisted reproductive technologies in these two paragraphs. Okay, collect the information and prepare a notes of that. I didn't prepare because it's already present in your textbook. It will be repeated. Okay, fine. Now here in my previous video of STDs, I have asked one question. All RTIs are STDs, but all STDs are not RTIs. Okay, for this I have got few comments, and those few comments which have been got, all are correct. No wrong answers I have got. I have got all the correct answer, which means all are interested to solve the questions, and they are knowing about the information what has been given in the textbooks. Okay, hepatitis B and AIDS are not infections of reproductive organs, which means these two are not RTI. Okay, reproductive tract infections are there, but these two are not RTIs. They are not infections of reproductive organs, though their mode of transmission could be sexual contact. These two are not RTIs, but they comes under STD. Because why they comes under STD? Because they are getting transferred from through sexual contact. Okay. All other diseases which I gave a list of a list of STDs like gonorrhea, syphilis and all other diseases are transmitted through sexual contact, and also they are infections of reproductive tract. Okay, they are having infection in the reproductive tract, that is they are in the reproductive organ. But these two, they are getting transmitted through sexual contact, but they are not having infection in the reproductive organ. So these two are not RTI, but all others are RTI and STDs. All RTIs are STDs, but all STDs are not RTI. How many reproductive tract infections are there? They comes under sexually transmitted diseases because they are transmitted through sex. But all STDs are not having reproductive tract infection. Hope you got the answer. Okay. Now, in today's class, what I have discussed, in that I am having one question. Please clarify my doubt that gametes are transferred in fallopian tube. Why not in uterus? Okay, already I have given an answer, but then I want the answer in the comment box. Please comment it so that you can I will come to know whether you are able to understand or not. Gametes are transferred in fallopian tube. Why not in uterus? This question I am giving here. Okay, fine. If you have any doubts, you can comment it in the comment box. And this was the last topic of infertility. Last topic in reproductive health called as infertility, and this chapter carries 5 marks. Reproductive health, it carries 5 marks in terms of exam. All the topics are very important and very easy to understand and very easy to score, so don't try to skip this chapter. Okay, so this infertility, very important. Infertility, what is infertility, what are the reasons of infertility, ART, very important. Okay, so all information you should know. And in my next video I'm going to discuss about principles of inheritance, that is genetics and evolution, next unit. And that unit is also very interesting and very interesting, and I'm going to come with the topics with the principles of inheritance and variation. And this unit which has been completed, that complete unit has been finished. If something I have missed, please comment it in the comment box. And this unit carries 25 marks, and the next unit also carries 25 marks. Both the units are very important. As we find that the CET exam has been completed, you can collect the question papers and try to answer the questions from these 4 chapters at least. What questions have been asked? The questions has been asked only from NCERT textbook. You can see, you can search and go through your textbook line by line and you will get the answer. Okay. Till then, stay safe, stay healthy.